Thank you everyone for joining. As been introduced, my name is Richard Lee. I'm the Director of Core Technology at ACDLabs, and for the next 15-20 minutes or so, I'll be discussing some of the challenges that scientists face, especially in deformulation workflows, and how some of the tools that we've developed at ACDLabs using the Spectris platform can address some of these bottlenecks. So in the last little while, we've seen a lot of consolidation and contraction of analytical labs. These labs tend to support a number of different types of departments and workflows. And so the mass spectrometerists now are challenged to do more with less. You may have more responsibilities and different responsibilities than you had originally been tasked with. The instruments themselves may also be used for different types of applications. So it's something that we've noticed, and we've also tried to address with software solutions. Some labs may actually go with, they may try to throw money at the problem in the sense that buying a higher resolution instrument or an instrument with a faster scan time or other features may address their bottlenecks. And in some cases they do, but more often than not, it will actually generate more data that the end user will actually have to sift through and analyze. And we also know that some of the vendor software that comes with the instruments may not be sufficient enough for the complex samples that the users are actually analyzing or tasked to analyze. Right? So we offer an alternative that offers an intuitive software package that complements some of the experimental approaches that you may have. So what is the informatics platform? What should it do? Right? So an advanced informatics solution should include the ability to support a diverse set of data formats. Now, in most labs, there are a number of different instruments from a number of different vendors. It's not like the 25 years ago where a lab may have been dedicated to a specific vendor. Now it's a wide variety of instruments that includes a wide variety of software that the end user has to be an expert at. The system should also support easy workflows. So the samples themselves are complex. The software doesn't have to be complicated. The system should allow for the data to be reviewable in the interface. So you should be able to view the chromatograms, mass spectra, UV spectra as well. Once the data has been processed, there may be new chemical entities that might be discovered that you had not anticipated. The data should be able to be reincorporated into the database for further downstream workflows. And the data and the results themselves should be shareable. So you should be able to communicate your findings to your other colleagues or other labs. So for us, one of the main core competencies is that our Spectris platform is vendor agnostic. So for the last 25 years or so, we've worked with a number of different vendors, all the major vendors, and we're able to support their data formats natively. For example, if you have a Waters QTOF instrument, the .raw file, you'll be able to simply drag and drop that entire data file directly into the Spectris interface, and you'll be able to visualize that data. This goes the same for if you have a thermal trap system or a Shimadzu TOF system. You just simply drag and drop those data files within our interface. And it doesn't matter if it's low res or high res, or if it's data dependent, or an all ions type acquisition. We're able to support all the variety of data within our interface. So how does the data actually get into the Spectris platform? Okay. So we have actually a number of different ways that we can pull the data into our interface. So we have the ability to just simply drag and drop. that data file. So again, if you just have a .d file for Magellent, you can just simply drag that data file into the interface and it'll just open up. You'll be able to see what you would see normally in Mass Center or ChemStation. You'll be able to see the chromatograms, you'll be able to see the mass spectra, any MS spectra, UV spectra as well. If you have a high volume of data that is being generated, you have the ability to sweep that data into our infrastructure. So again, we have the ability to configure what we call automation server that allows us to just simply scan for a particular instrument or data source, and that can be pull back the data when it's prepared, and then it gets converted into our format that can be viewed by the end user. We also have the ability to perform data processing routines from completely manual to semi-automated to completely automated. And what I mean by manual is that the end user pulls the data directly into the interface. The user then can simply work through the icons and process the data. Or if you have a batch of data that you may want to process with the same processing routines, you can configure that within the Spectris interface. But also, if you are the end user and wants to configure the system because you have such a high volume of data to be fully automated, meaning that the system will just sweep in the data, perform the data processing routine, generate a report or a review of the data or the results, you have that option as well. So all the data processing routines and workflows that I'll be discussing in the next few slides can be performed automatically using automation server to be performed in the background silently. So for us, when we say deformulation, it doesn't necessarily mean that it is a sample from an environmental or food and beverage. The provenance of the data set doesn't really matter. It can be forensics, it could be a data set from a complex mixture from the synthesis group, but it's the ability to isolate a particular component and being able to identify that particular component itself. So for us, there are three major parts for a deformulation workflow. First is to isolate the actual component. Can we isolate the mass spectral features into to a specific component or peak and create a component mass spectrum that includes molecular ion, any adducts, any fragment ions, etc. And to be able to isolate that from background noise. And then can we interpret that component mass spectrum? So again, can we estimate the molecular ion? Can we identify any adducts? Can we identify any multimers, multi-charged ions? Once we've isolated the component, the next step is identifying that particular component. And that may include a spectral search, whether it's MS1 or MS2 against your own database that you've created, or if it's a commercial database that's available. If you have no spectra that you can search against, you may want to perform a molecular ion search. And you have maybe a number of different metrics that you use for filtering the results. So again, if you have a molecular formula or a chemical structure or a retention time, you can use that as well. And then once the data is processed, you may have a new chemical entity that you may want to add to the database for workflows later on. And the data that you've processed can be leveraged for future use. And also to be able to generate reports. So this is key. How can you share your results, whether it's via a PDF or Word document? You may want to just export relevant key data. So again, just a set of metadata and the results table that gets pushed into an ELN record. Or it could be a specific data format with this information that gets pushed into a data warehouse. So those are the key steps within the deformulation workflow. And I'll go through each one of these in a bit more detail in the next few slides. So the first part of the data processing is really the component extraction. How do we go about actually isolating a component? How do we group common mass spectral features into a component mass spectrum within a defined retention time range? So in order for us to do this, we've actually developed an algorithm that we call IntelliExtract, and we've leveraged some technology called ion threads that allows us to group these XICs together to create a component XIC and a component mass spectrum. As you see down here in the image below, this is an image of what the interface will look like. It's a bit scrunched, but you can see the chromatogram as well as the XICs, the overlaid XICs. You see a pure component mass spectrum in the bottom left that has the molecular ion as well as any of the adducts that are labeled. You see dimers. You see also be able to label multimers, any type of multimer, multiple charge species as well. We also have the ability to extract out the MS2 spectra of any component that we've identified. And again, it's not just a single MS2 spectra. If you have multiple collision energies, we're able to extract all the MS2 spectra for any of the collision energies that you have acquired on. But once you've actually componentized, we have a number of different routes to go about actually identifying the component. First is what we call IntelliTarget. This is a very simple, straightforward algorithm. So it is a very small subset, and as the name suggests, it is a targeted approach. Next is something called IntelliExtract Compound Recognition, or IXSCR. So this is for those known unknowns where you have a spectral search that gets performed along with a number of different filters. If you have no spectra though, but you have a database of structures, you have the ability to perform certain searches against the molecular formula of the database, but we actually incorporate a number of different strategies within something called MS Structure ID Suite. That includes a local version of the PubChem database, so you'll have access to millions of structures. But also we perform an in silico fragmentation that we can assign to the MS2 spectra to give you a bit more verification. So I'll step through each one of these as well. IntelliExtract is our most straightforward type of identification algorithm. You have a specified list, that's the input. Within that list, you have either a reference mass, you could have a formula, as well as you could have just a structure. So again, if you have a structure, the system will automatically calculate the formula and provide the reference mass. You may also have a retention time that gets associated. So again, this approach is for data sets that you have the same method and you have a similar set of targets that you want to look for. Once the targets are found, the chromatograms and spectra are annotated with the appropriate compound names. Next is what we call IXSCR. So IXSCR is a spectrum search algorithm, but it's just simply more than a spectrum search. It will perform searches and filtering based on the component mass that's identified against a database record. So if you have a structure or if you have a precursor value, but with no structure, it will search against that. It will return hits based on MS2 spectra or MS1, if that's all you have. And it will perform a mirrored comparison, as you can see here. And again, you can filter it based on, if you have different collision energies, a specific collision energy value. Okay. But if you happen to not have any spectra at all, you really only have a database of structures. So again, let's say you have access to PubChem, or if you have a database of proprietary chemical structures that you want to search against. Well, we have the ability to perform this type of search as well. So again, once you have an isolated a component with a molecular ion, you can perform a search based on that potential molecular ion and reducing the number of hits based on mass accuracy. You can limit the chemical formula range as well. It will then perform a theoretical isotope pattern match against that of the experimentally found component. You can include substructure inclusion-exclusion lists. So again, if you have an idea of the type of substructure that it might have, you can include that as part of the filtering mechanism. But we also have the unique ability to perform in silico fragmentation. So we will take all your potential hits. So again, you could have specified the top 5 hits or the top 10 hits. And it will perform an in silico fragmentation on an MS2 spectrum. It will assign the fragments to that spectrum and provide a spectrum assigned value that you can use further down the line to isolate that particular hit. But as you're processing your data, you may not come back with a hit because it is truly a unique chemical entity that you've discovered. It may be something that's unknown to you, but you know that you may want to use this for searches later on. You have the ability to update your working database with this particular record. So again, if you have a component, you just simply have to click on a single icon that allows it to be updated to the database for future use later on. So again, if you have other data sets that you may want to process, you can use this newly added record as part of that database search. It will store your chromatograms as well as any mass spectra that are associated, all the metadata that's available from that data file itself. You can update the actual record with a chemical structure if you've identified it later on. So again, it's a very flexible database, and this is one of the keys to growing your database for future use in any of your workflows. So once the data is processed, you may actually have a wealth of mass spectral information, chromatographic data, structures that you will want to report. Right? You'll need to be able to share this information with all your colleagues or other departments. And this might be just through a simple PDF document. You may want that PDF document to be circulated or a Word document of your choice. You may be preparing a much larger report that you can just simply copy and paste parts of your results into that document. Within the Spectris platform, you have that ability. So you can create a customizable template that can create a PDF or Word document. But let's say you also want to share this information electronically. You can simply have that database of your results to be shareable, or you may want to extract out a part of that results, part of that data into an ELN or into a data warehouse. So you can do that using our API. So if you wanted to only just extract out a subset of the results, you have that ability. So it is a fairly rich API that you can manipulate or use to extract out specific sets of information in specific formats, depending on your third-party application. So I hope in the last 15 or 20 minutes, I've been able to show you what ACDLabs has to offer in terms of addressing the challenges that you face in the deformulation workflows, from importing the data through processing it, and then actually being able to share that information with your colleagues. If you have any questions, please feel free to ask. Otherwise, you can always contact us for additional information. Thank you.